Hi students, welcome to Vivid Botany. We have a concept of nucleosome here. Nucleosomes appear like beads on string-like structure in nucleus. See, for example, I draw a small diagram of nucleus here. Let us suppose this is nuclear envelope, what we call as the karyotheca. This is karyotheca, that is the nuclear envelope having outer membrane and inner membrane. It is discontinuous, having pores. So inside this nuclear membrane we find the chromatin reticulum. This is chromatin reticulum. On this chromatin reticulum we have small bead-like structures. The small bead-like structures are called as nucleosome. So they are called as beads on string. So these beads on string are nothing but the nucleosomes. So a nucleosome diagram I drew here. It's made up of 8 histone proteins. A nucleosome is made up of 8 histone proteins, protein molecules. So they include here H2A of 2 molecules, H2B of 2 molecules, H3 of 2 molecules, H4 of 2 molecules. Total it's made up of 8 molecules. So it's called as octamer. One nucleosome is called as octamer. Outside that nucleosome, another protein is present, that is H1. So with H1, it is nanomer. So this is interlocking protein. Interlocking protein means this protein will help locking and unlocking of the DNA. The red colored thread that I drew the diagram here is nothing but the DNA. This DNA is of total 200 base pairs. So this DNA wraps around the nucleosome twice, which is called as super helix. The DNA which wraps around the nucleosome twice is called as super helix. So 8 histone proteins will make up one bead or one nucleosome. So nucleosome is called as octamer, and outside that nucleosome H1 protein is present. With that it is nanomer. Then why histone proteins are specifically make the nucleosome? What is the importance of histone proteins? These histone proteins are made up of basic amino acids. They are made up of basic amino acids. What are basic amino acids? These are having the amine groups or basic charges at their ends, like lysine and arginine. Lysine and arginine. These are two basic amino acids by which histone proteins are made up of. And we know that DNA is acidic. Because of the phosphate, DNA is acidic and DNA is negative, whereas histone proteins are basic and positive. So what happens here? The DNA being negative wraps around the positively charged histone proteins and gets electric neutrality. The electric charge of DNA gets compensated by association with the histone proteins. So DNA becomes more stable as it has to act like genetic material. It needs to be more stable. So this is the nucleosome concept, which appear like beads or string in the chromatin reticulum. This is chromatin reticulum, the word coined by Fleming. This is nucleoprotein fibers present inside the nucleus. So these beads are nothing but the nucleosomes. Now let us see the question here. Which one of the following statements about histones is wrong? Histones are organized to form a unit of 8 molecules. This is very much correct. The pH of histones is slightly acidic. This is wrong. They are not acidic, they are basic. So second one is the answer. Histones are rich with amino acids, lysine and arginine. It is correct. Histones carry positive charge on their side chains. This is also very much correct. Remember always, acidic is negative and basic is the positive. So it's about histone proteins. Hope the diagram is very well understood to you. And once you have understood the concept, any type of question that can be asked, you can do that. For example, I'll ask you here one question: how many H2 proteins are present in one nucleosome? How many H2 proteins are present in one nucleosome? You may say it is 2, that is wrong. H2A, H2B, total 4 proteins are present of H2. How many H3 are present? 2. How many H4 are present? 2. How many H1 are present? Only one. That two outside the nucleosome. It is called as extra nucleosomal protein. H1 is called extra nucleosomal protein. Why it is extra nucleosomal protein? Because it is present outside the nucleus. So this is about the nucleosome concept, and please subscribe my channel Vivid Botany. We will be tackling and discussing all the questions of NEET 2021 and as well as NEET 2020 also. So thank you very much. Let us see the next question in our next video. Thank you.